Hello, everyone. Welcome to the second podcast for Chapter 20, Day 20 on biotechnology. In this podcast, we're going to focus on Objective 3, going a little out of order. And in this objective, we're going to talk about gene therapy. This part of your chapter goes pretty deep, but there's just some really basic information you need to know. So this will be a pretty short podcast. Okay, so let's go ahead and get started. In the first podcast, we talked about how we could take a bacterium. So let's draw a bacterium here. And if you remember, we put our plasmid in here with the human insulin gene in it. And then we took that and we put it in a big flask with media in it, like so. So we put it in here. It grew as it was growing through the process of transcription and translation. It made the protein, human protein insulin, and then we took the insulin out of here, out of the flask, and we were to package up into vials of insulin and provide that for humans to treat their insulin. This turns out to be an incredibly efficient way to mass produce insulin. In fact, many of the drugs we use today, this is how it's produced, particularly the human forms of certain proteins that are injected. Often they are made in bacteria or other microorganisms like yeast. Now, one big difference between this and the next strategy I'm going to talk about is that this is strictly a treatment. This is not a cure. This is a treatment. This other thing I'm going to talk to you today about is called, in fact, let's draw a line here. It's called gene therapy. And you've probably heard about this. This is a very common word we hear in the media. And in general, we haven't had a lot of success with gene therapy. We've had a few pieces of success and a lot of promise, so it's there. We just haven't realized it quite yet. All right, so gene therapy, well, let's back up. So what we called up here with gene cloning, in fact, why don't we put gene cloning here? In gene cloning, we're giving the person a protein. We're giving the person the human insulin protein to treat them. In gene therapy, our hopes are that we will be able to give somebody the DNA, the gene, the insulin gene, and that this could be a cure. Cures are better, right? Because you don't have to keep taking the treatment. Maybe not quite as profitable for a pharmaceutical company, the cure, but certainly it's better for the person. All right, so how do we do gene therapy? Well, we're still going to use the human gene, and we're still going to make it the same way using PCR. So here's our human gene. And what we're going to do is we're going to place it in a virus. And often when we hear the word virus, we immediately think this has to be something bad. But it doesn't have to be. In this case, it's likely not. So we're going to clone that gene into a virus. And then this virus is going to be packaged up into some sort of what we call a capsid. Just a membrane around it to help protect the DNA. The idea now is to be able to give this virus and place this into humans. So here's our human. Of course, the size ratio here is completely off. Humans are much bigger than a virus. But we're going to take this virus and place it into the human. Now what happens is within the cells of the pancreas, and our hopes are that we can target this virus to the pancreas. So this virus goes to the pancreas. So now from this pancreas, let's take out a cell. Let's put it down here. I apologize for the scribble. I started drawing something that I didn't like, so I erased it or scribbled over it. So here's a cell from the pancreas, right, like so. And in the nucleus of this cell, we have DNA. So here's the human chromosome right here. And remember, this is a person who has diabetes type 1. And here's their gene of insulin, but remember, it's mutated. When it tries to make a protein, it doesn't make insulin, the human insulin protein that's going to help this person live. It's mutated, so it doesn't work. So the idea then is that this virus, and when this virus enters the cell, it sheds this outer coat here, so just the DNA portion is in here. And so it's going to be in the cell, in the nucleus here. And here's our viral DNA, and here is our human insulin gene. They're right by each other. Human insulin gene, it's the healthy copy, it's the copy that the humans need in order to make functional insulin. And what happens here is that we have recombination between the DNA on this viral DNA and the DNA in the human cell. And I'm just going to draw it like this, because it's crossing over. We say it's crossing over, so we'll show that by having an X. But really what's happening here is that this good piece of insulin, the proper insulin gene, is now moving down here and replacing this mutated copy. And so what happens here is, after crossing over occurs and it moves in here, we'll have our chromosome here where the insulin gene should be. And the mutated copy, the one I drew here in black, is now gone. It's been gotten rid of. And we've replaced it with the human insulin gene. So what has now happened is we now have created a cell. And in theory, we have created a whole bunch of cells. We're just showing one cell here. When we infect somebody with this virus, we infect them with a lot of the virus. And so now we have many cells in the pancreas that have the correct form of the insulin gene. And now this insulin gene in the human will start to make messenger RNA through transcription, then eventually start to make protein, the protein insulin, through translation. And now it's made this insulin protein itself, right? We're not giving him or her the protein. They're making it themselves. And so now in theory, this person has this new gene in them, in their pancreas, the new insulin gene in their pancreas, and now they're going to be able to make the proper insulin that they need. If this works, what would happen is now this person would no longer need to have daily injections because they're making their own insulin. So it's a really cool idea. So you might have noticed that as I was talking about this, I kept saying things, if we could do this. And the fact of the matter is, this has not yet been done in humans. We have not successfully been able to do this. But scientists have been able to show that it's possible by showing this in other organisms. So for instance, in mice that scientists have made diabetic, we know that they will respond to gene therapy in this way and essentially cure themselves of diabetes. So the scientists know that this is possible, but more tests have to be done, and it has to be proven in other animals before we can begin some initial trials. So hopefully someday in the near future, something like gene therapy to cure diabetes will become a reality. We're really close. All right, that's all I have for this podcast. In the next podcast, we're going to start talking about stem cells and their use in diabetes, and we're also going to talk about cloning whole organisms. That's not necessarily related to diabetes. It kind of diverges away from our theme for these podcasts, but it's an important thing to talk about. All right, well, that's all I have for this objective. So if you have any questions, please let us know, and if not, we'll see you in class. Bye.